Farmers have been breeding fruit and vegetables for hundreds, if not thousands of years in an effort to maximise yield and get the tastiest product to markets as possible. But traditional plant breeding is a long, slow, laborious process requiring years, if not decades of work and thousands upon thousands of plants. Well, that may be about to change. And that's all due to a process called Crispr, a form of targeted gene editing that allows scientists to modify a plant's genome with ultra-high precision. Now, this can allow breeders to generate new varieties of plants with potentially beneficial characteristics at a much greater speed. And we're also using CRISPR here in the University of Birmingham's School of Biosciences in order to create specific mutations in this, the model organism Arabidopsis thaliana, in order to understand more about a process called meiosis. But how can we tell if we've created a mutation in a plant's genome? After all, DNA is incredibly small, impossible to see with the naked eye. Well, one method we could use is a special form of chromatography called gel electrophoresis. Now, let's say I have a plant gene. Now, using CRISPR, I've purposefully taken a large section out of the middle of that gene, so my mutated gene will be much shorter than my non-mutated gene. Now, using gel electrophoresis, I'll be able to identify which plants do and don't have this mutation. So for electrophoresis, I have the following equipment. I have a gel made of agarose with small wells at one end. The gel is sat in a tank surrounded by a special buffer solution, and an electric current can be passed through our gel in order to separate charged substances. Here we're separating DNA, which is negatively charged and so should migrate from the negative electrode towards the positive electrode. In these tubes I have some DNA samples from plants which may or may not contain my mutation. What I need to do is add a small volume of each DNA sample to a well in my gel. And then apply an electric charge. The gel is essentially formed from a tiny meshwork of cross-linked polymers which act as a molecular sieve. As such, the smaller molecules will move more quickly through the gel, whilst the larger molecules will get caught up in that mesh and travel more slowly. The gel also contains a special stain so that when we finish with our electrophoresis, we can visualize our DNA under ultraviolet light. So we end up with a number of bands that have run different distances from the top of our gel. The bands that have run further are from plants containing the deletion mutation, which generates a shorter gene, and the bands nearer the top are from plants without the mutation, so they have the normal length gene. This process of molecular sieving also applies to different types of chromatography, such as thin layer chromatography, which you may be familiar with in the classroom, where substances are again separated based on their molecular size or charge. So, we've demonstrated how gel electrophoresis can be used to analyse the DNA from plants that have undergone CRISPR-Cas targeted gene editing. This technology has the potential to revolutionise plant breeding, allowing breeders to produce bigger, better, hardier crops at a much greater speed.